The clock strikes 2 p.m. and we are live here as Scandion Oncology joins us to give us a research and development update. And standing beside me is CEO Francois Matelet. Welcome very much.
Thank you very much.
You're here to present, of course, and I'll return to ask a few questions. And I will also take this opportunity to remind our viewers that if they have any questions during this broadcast, not to hesitate to ask them in the live chat below. I will be keeping an eye on it. Now, Francois, go ahead.
Thank you, and welcome to this research and development update, which is an update of the earlier version we had a few months ago in March of this year. And this is also an investor day. So as you said rightly, you will have an opportunity to ask all questions that you may have to us. Today, this is a disclaimer that we will not go through in details, obviously. Today, we have gathered the whole management team in order to present where we are as a company, and even more importantly, where are we going. And then, after me, there will be our Chief Scientific Officer, Jan Stenvang, going through the mechanism of action, in details. Then we will have a little bit of a surprise to you today because Professor Tabernero, who is a leading oncologist, leading in the medical oncology field, especially in GI, gastrointestinal area, has accepted to record a video to give us and to give you his perspective on Scandion Oncology and where we are with regard to our data. Then we will have our Chief Medical Officer, Lars Damstrup, going through the data as well. Then we have, this is also a significant event for you, we have our CFO, Chief Johnny Stillo, to go through the right issue. And then I will conclude this Research and Development Day at the very end of all presentation. So, having said so, this is the live picture of all management team of Scandion. I would like to mention a few things that we have very recently strengthened very significantly the board of the company by adding 2 board members, Professor Pfeiffer from Denmark and Professor Michel Ducreux from Gustave Roussy in France. And these 2 leading experts in GI are certainly a great addition to our board, and so we are very well equipped, from a capability perspective to move this company forward into the right direction. And having said that, I will now pass the word to Jan for going through into the cancer drug resistance. Yeah. Please.
Thank you, Francois. Good afternoon, everybody. It's a pleasure to be here and present for you. Obviously, we are a company focused on drug resistance, cancer drug resistance. And why is that? That's because on a global scale, 20 million people almost are diagnosed with cancer every year, and 10 million dies every year from cancer. And these numbers are unfortunately increasing. And as you can see in bold text, the major problem is that up to 90% of cancer deaths are related to resistance against the current treatment, and there are no drugs that are actually available to counteract drug resistance in cancer. So we think there's a huge unmet medical need here, and everybody actually agrees on that. Our approach to that is that we have a drug that targets 2 different targets. I will come back to that in a moment. One of the major ones is efflux pumps, such as ABCD2, as mentioned here. That's a very common mechanism for both acquired resistance and intrinsic resistance. Intrinsic means that you are resistant as the first time you get the drug, you're simply resistant. Acquired means that the resistance will be acquired during drug treatment. So that is the problem that we are trying to address. Obviously, these efflux pumps are not the only thing that can contribute to resistance in the patients. You can see we have listed 3 very broad categories here. One is that drugs may be effluxed from the cancer cells, so they won't work inside the cancer cells where they're intended to work. Second one is that something changes within the cell. There's many things that can change in a cancer cell, and therefore the drug may be less effective. The last one is that maybe the drug does not enter the cancer cells as well. However, we are focused on number one, the drug efflux mechanism. So I'll take a little bit of time to go through in some details and give a highlight presentation of what it means in the end, because SCO-101 is a very special molecule. There are no other molecules on the market that is doing what SCO-101 is doing. So therefore, it's very important that we go through this mechanism of action. How does SCO-101 work? It has two effects, and here is just a highlight. I will go through it in details afterwards. One is an effect directly on the tumor cells, or the cancer cells in the tumor. And these cells, if they're resistant due to ABCD2 expression, so that means they have a high level of an efflux pump, we can block that. The other one is actually outside the cancer cells, and that's a plasma effect where you can increase the chemotherapy levels in the blood. Okay, let's take them one by one. So ABCD2 first, which was the efflux pump. First a cartoon to kind of illustrate the point here. So in a relatively normal setting, if that exists, you add chemotherapy to the patient. The chemotherapy will accumulate inside the cells and hopefully kill the cancer cells. That is luckily the case very often. However, resistance is unfortunately very often emerging, and if it's by ABCD2, then you have the picture here, and you can see that the chemotherapy is expelled from the cell, meaning that there's less chemotherapy inside the cell and it's less likely to kill the cancer cells. So that means resistance. However, SCO-101 can block this efflux, and thereby chemotherapy will accumulate inside the cancer cells and hopefully kill the cancer cells again. So that is the overall take-home message. I have a few data to support these points. So what you see here is a little bit of a preclinical data from our lab. On the left-hand side, we have made activity assays for ABCD2 and two other relevant efflux transporters. And if you look at the y-axis, you can see that this is the activity of these transporters. So 100% means that it's fully active, 0% means that it has no activity. And then on the x-axis, we have increasing concentrations of SCO-101. So what you would expect for such a curve, if SCO-101 inhibits such an efflux transporter, is that the curves in a dose-dependent manner would go down. And this is exactly what we see. And furthermore, we can see that the ABCD2 is inhibited very early. That means lower concentrations of SCO-101 is needed to inhibit ABCD2 compared to the two other ones. You can describe that by the value named IC50. So that means the amount of SCO you need to have 50% inhibition. And you can see it's more than 100-fold more selective for ABCD2 and than ABCB1 and ABCC2. So that means we have a selective and potent inhibition of ABCD2 in this lab experiment. Of course, we're also interested to see if the other point is true, that if you use SCO-101, can you actually increase the active level of chemotherapy in the cells? So this is the right-hand slide demonstrating that. Again, you have the amount of chemotherapy on the y-axis. We made it radioactive, so we can measure how much there is. This is the y-axis, the amount of chemotherapy. On the x-axis, you have the concentrations of SCO-101. And what you can hopefully appreciate is that when you use higher and higher concentrations of SCO-101, you will have an increase in the curve, and that means you will have more chemotherapy inside the cells. And these are, of course, ABCD2 positive cells. So these two simple pieces of data kind of supports the notions that we have ABCD2 inhibition and accumulation of chemotherapy inside the cell. So we also developed some resistant and cancer cells that have a high expression of ABCG2. You can see a Western blot on the right-hand side that shows how much protein is present, and you can see that to the right-hand side you have the SN38 res, it's called. That means the resistant cells. They have a high level of this efflux pump. That corresponds to the two red lines. The red lines is just the chemotherapy alone, and what you see on the y-axis is the viability of the cells. So that means if it's 100%, all the cells are alive, and you have 0%, none of the cells are alive. So it's an expression for how many cancer cells are alive. So when you take the red solid line, you can see that at very high concentrations of chemotherapy, you can even get an effect on the resistant cells. If you then look at the red dotted line, this is what happens when you have SCO-101 present together with the chemotherapy. It moves a lot to the left, basically all the way down to the sensitive cells. So that means when you add SCO, the chemotherapy works again. So that's a proof of concept from a resistant cell line that you can make the chemotherapy works again. Okay, so that was ABCG2. The other target is a liver enzyme that is present in all human beings. Its name is a little bit complicated, but it's UGT1A1. So on the left-hand side, you can see that in the cartoon above the graphs, this is what happens when you give irinotecan, which is chemotherapy that is used for colorectal cancer. When you give that to a patient, it will be converted into SN38, and that is the active chemotherapy. Normally that is inactivated by this UGT1A1, and you can appreciate the triangle which represents SCO-101. It blocks UGT1A1, and we have increased levels of chemotherapy. So once again, we wanted to investigate that a little bit more in details. So on the left-hand side, the graphs as you see here is on the y-axis is the activity of the UGT1A1 enzyme, so how active is it? And on the x-axis we have the SCO-101 concentrations. And we have used two different substrates for UGT. We have used beta-estradiol, but we have also used the real active chemotherapy, which is the blue line. And what you will appreciate that when you increase the amount of SCO-101, you have a dose-dependent reduction of the activity of UGT1A1. So it clearly demonstrates our point. There's also other family members of UGT, and we have investigated 6 of them. This is what you see to the right-hand side. And what you can appreciate is that this IC50 value that I mentioned before, where you have a concentration that gives half activity, it's 0.1 for the active chemotherapy, and it's much, much higher for the other UGTs. So that means that SCO-101 blocks UGT1A1 from converting active chemotherapy into inactive chemotherapy, and it's selective for UGT1A1. So I'd like to try to put that into context where we hit both ABCG2 and UGT1A1 by a simple cartoon. So normal setting again, if there's anything normal about cancer, you add chemotherapy, it goes inside the cancer cells in the tumor and hopefully kills the chemotherapy. But also outside the cancer cells, you have UGT1A1, which inactivates the chemotherapy. That should be sufficient to kill off a number of cancer cells. However, if resistance emerge, as I showed you before, the ABCG2 will be able to remove chemotherapy from the cell, making it less likely to work, and still UGT on the outside will inactivate the chemotherapy. So that's a problem. Our solution with SCO-101 has the potential to hit both the ABCG2 and the UGT1A1. As I showed you before, it will block ABCG2, increasing the level of chemotherapy inside the cell, so we're hitting the cancer cells from inside. However, we will also hit outside by hitting UGT1A1, making larger concentration outside the cancer cells, so we will have an impact from chemotherapy outside and inside. So that's a double hit mechanism, and we have clinical data from our studies, CORIST studies, to support this mechanism, and Lars will briefly allude to that a little bit later in his presentation. So I hope it's clear that we have a new type of molecule. No drugs on the market that we know of inhibits ABCG2 and UGT1A1 at the same time, and none of them has, there exist no drugs with this mechanism to prevent or counteract anti-cancer drug resistance. So we truly believe that we have a novel mechanism of action which could eventually be first in class. So thank you very much, and with that, over to Lars.
Thank you. Before I begin with my presentation on the clinical update, I'd like to turn it over to Professor Tabanero, who was very graciously accepted to do an interview to tell his view about our drug and how we see it in the future and cancer resistance, etc. So before I continue, could you play the interview, please?
Professor Tabernero, you are a member of the Clinical Advisory Board at Scandion. I thought we could begin by an introduction of your background and specialty.
Thank you very much. I'm José Tabernero, a medical oncologist by training. I'm professor of medicine and head of the medical oncology department at the Vall d'Hebron University Hospital in Barcelona, and also the director of the Vall d'Hebron Institute of Oncology, that is a research institute that covers all cancer research in this institution. I have been a member of the executive board of the European Society for Medical Oncology, ESMO, and have served as the ESMO president for the term between 2018 and 2019. Moreover, I have been appointed as a member of several educational and scientific committees, not only from ESMO, but also from the American Society for Clinical Oncology, the American Association for Cancer Research, and other professional societies related to the field of cancer. My special focus have been treating patients with gastrointestinal malignancies, in particular colorectal cancer, and also in the creation of the phase 1 unit, the early drug development unit at our institution, that is one of the largest in Europe.
Colorectal cancer being one of your specialties, could you tell us a little bit more about the condition and the greatest challenges in treating patients?
Sure. First of all, colorectal cancer actually it's a very frequent disease. Depending on the countries, it ranges between the first position and the second position, and mainly because it affects both men and women. So anything that we do actually to increase the survival in this population of patients, especially in those that have advanced or metastatic disease, is really very important in terms of numbers, but also in terms of societal needs. It's very important to mention that metastatic colorectal cancer continues to be an unmet clinical need for the management of these patients. Right now, actually, we have well-established up to 4th lines of therapy for metastatic disease, combination of different chemotherapy schedules, FOLFOX with oxaliplatin, FOLFIRI with irinotecan, a number of targeted agents, either related to inhibit the angiogenesis process, like bevacizumab, aflibercept, and even ramucirumab in some countries, but also targeting the epidermal growth factor receptor, like, for example, cetuximab or panitumumab. Also, we have drugs for the more refractory setting, like the combination of trifluridine tipiracil plus bevacizumab, well established in the 3rd line setting, and also very recently fruquintinib, a small molecule that inhibits angiogenesis as well, and that has been recently approved for the 4th line setting. But as you can see, we have several lines, but still patients, even they respond to these therapeutics. At some time point, they progress under therapy. And the reason for that is because drug resistance appears at some time point. And this is really a major problem that we have with all these cytotoxic drugs and biological agents. So it's very important that we work on that. And actually, one area of improvement is to revert as much as possible drug resistance when it appears. So we need to improve, several areas of treating patients with metastatic colorectal cancer. And one area, as mentioned, is trying to revert drug resistance.
Is that the challenge that the lead candidate SCO-101 from Scandion seeks to address?
Exactly. So SCO-101 is a well-designed drug that actually targets two different enzymes. The first one is what we call an efflux pump. This is a kind of enzyme that tries to excrete any kind of drug, in this case a cytotoxic agent like irinotecan, outside from the cell. And what we do see in cancer patients is that this efflux pump called ABCG, it's very often unregulated in patients with colorectal cancer, specifically in those that have resistance to some drugs, including irinotecan. What actually happens with this is that this upregulation of this enzyme ABCG causes a lower level of chemotherapy inside the cancer cells, and therefore the chemotherapy drug works inefficiently to kill the cancer cells. So by targeting these efflux pump enzymes like ABCG2, in this case with this compound SCO-101, the chemotherapy drug, in this case irinotecan, will accumulate in the resistant cancer cells and ultimately kill the cells. This is the first mechanism of action of SCO-101. The second one actually is targeting one of the multiple metabolic degradator enzymes for irinotecan or SN38, that is the active drug of irinotecan. So these drugs, irinotecan and SN38, are degraded through a glucuronidization process. And in this process, one enzyme that is present in the liver is very key. This is 1GT1A1. Actually, this enzyme 1GT1A1 inactivates SN38, and therefore, if we have a drug like SCO-101 that is able to inhibit in part 1GT1A1, this may also cause an increase of chemotherapy, both in the liver and in the circulation, which actually will lead to an increased exposure of the cancer cells from the outside. So just to sum up, SCO-1 is a well-designed drug that by dual targeting of ABCG2 and UGT1A1 is able to increase the levels of both internal and external irinotecan exposure to the cancer cells, and thereby hopefully killing more cancer cells.
And recently we actually got data from the part 3 of the CORIST phase 2A trial. Could you give us your view on the result of this data and why it's important?
Yeah, actually we have data from different CORIST studies because CORIST part 3 is the more recent one. But actually I would like also to mention that we have CORIST 1 and CORIST 2 or part 1 and part 2 that actually were critical studies to define the recommended phase 2 dose for SCO-101 in combination with chemotherapy, in this case FOLFIRI, but also studying very preliminary the efficacy in patients that were refractory to several lines of treatment, including irinotecan-based chemotherapy. But coming back to the question that you did, focusing more on CORIST part 3, actually this was a very important study because in this study the dosing schedule was redefined based on the previous pharmacokinetic data observed in CORIST part 1 and part 2. So two new schedules were designed, one with a 6-day administration and another with a 4-day administration of SCO-101. So basically, this was important in order to define which was the best strategy to treat patients with refractory metastatic colorectal cancer. More importantly, early signs of efficacy were seen in this study. So, for example, in the cohort with the highest SCO-101 dose, that was 250 milligrams, in 6 patients, actually 2 partial responses were observed. And this is really very important. It's a biological proof of concept that SCO1 is working in the way that we expected. So SCO1 was able to reverse secondary resistance to previous administration of irinotecan-based chemotherapy. Moreover, if we look at the whole population that was included in part 3 of the CORIST study, the efficacy data was encouraging in a way that we observed a median progression-free survival of 3.8 months and an interim, because it's very preliminary, median overall survival of almost 8 months. But actually, this is really very important compared to historical controls. Also, we are working extensively on trying to evaluate whether there is any predictive biomarker that could even help more to define which are the patients that may benefit more from the administration of SCO-101 in combination with irinotecan-based chemotherapy. So we are looking at the importance of the different variants, genetic variants of one of the target enzymes, UGT1A1, for example, and this is work in progress, but actually I think that this is going to be probably provide important information to redefine a little bit more the population, and also other biomarkers that we are very actively evaluating. But perhaps to sum up, I would say that the data that we have from the CORIST studies, part 1, part 2, and part 3, have demonstrated the proof of biological activity, the proof of concept that SCO-101 is able to revert acquired resistance to irinotecan-based chemotherapy.
And what does that mean for future development of SCO-101, but also the CORIST study?
Of course, we need first an initial demonstration that we have an active drug with a recommended dose, and that it's able to prove the concept for what it was created, right? This is what we had with the previous studies of CORIST. Now, we are in the process of consecuting a randomized phase 2b study. Of course, we have always to demonstrate that this regimen, irinotecan-based chemotherapy in combination with SCO-101, is able to be superior to what we consider the standard of care. And this is the need for a randomization. So the randomized phase 2b study will be exploring at the same time several parameters, such number of patients, randomization, endpoints, and this has to be still discussed with external experts. Of course, this is going to be the first demonstration consistently showing that there is a superiority in efficacy. Later on, I think that the company probably will discuss whether they think that the data is good enough to move to earlier lines of therapy. So even in patients that are irinotecan naive. But first, we have to demonstrate in a randomized fashion that the combination of irinotecan-based chemotherapy plus SCO-101 is better than what is considered the standard of care.
I thought we could end by you giving us your final thought on this candidate being a combination therapy to chemo.
Absolutely. I think that with SCO-101, we have seen that we have a very promising drug candidate that can be safely administered in combination with irinotecan-based chemotherapy, that works as it was expected by this innovative dual targeting, and therefore be a first-in-class kind of drug that it's able to reverse secondary resistance to irinotecan-based chemotherapy. And of course, it addresses a very important clinical problem with a huge medical need.
Thank you very much, Josep Tabernero.
Thank you. It was my pleasure.
call, and I also like to thank Professor Tabanero for his insights into core-risk drug resistance and our future potential development program. Now, the next slide, please. I appreciate that many of you have been part of the core-risk journey for many years and know what we have been doing over the last many years. But just to highlight, we have conducted the part 1, which was a dose-finding trial. This has been concluded successfully in as much as we have defined the recommended phase 2 dose. Now, the part 2 was an efficacy assessment of SCO-101 in combination with FOLFIRI. Although we did not meet the primary endpoint of response rate, we are still thrilled about many of the other efficacy parameters, which I'll come back to later. Now, core-risk part 3, and also as Professor Tabanero mentioned, is an extremely important part of the core-risk trial, as it combines the data and the information that we have from part 1 and part 2, in as much as we defined, based on the pharmacokinetic parameters, we defined 2 new dosing schedules, which I'll come back to later. And the next slide, please. Now, on this slide is highlights of the data from the core-risk part 2 trial. So we did see successful tumor reduction, in as much as we saw about 20% of the patient population had a tumor reduction. However, this was below the predefined threshold of 30%. We did see an impressive median overall survival and a very good, and also a very, very long overall survival, which also impressed Professor Tabanero. We did see an exciting clinical benefit rate of 42%. And we also looked at the data to establish whether there's something in the data set that we could look further into, and this was bilirubin. I'll come back to that a little bit later, but this could serve as a surrogate biomarker for response. And the next slide, please. Now, these are very, very important data, which summarizes what we have seen from core-risk part 2. On the left-hand side is what's known as a Kaplan-Meier plot, which shows survival, both overall survival and progression-free survival. On the y-axis is the percentage of patients. So 100% means that all patients are alive, and 0 means that all dead or have had a progression. And on the x-axis is the time frame, in this case, days. And if you mentally put a line at 50%, and then for the solid line, this is progression-free survival, and this means that, or this shows that the progression-free survival is 2 months. And if you continue this line all the way to the dotted line, which is overall survival, then it's 10.4 months. Very, very impressive when you compare to historical data, which is in the ballpark of 5 to 7 months. There are one study, which I'll come back to, which also Tabanero mentioned. He mentioned the drug's name, but this was the trial called the Sunlight trial. And in this trial, there was a overall survival of 10.8 months, so completely in line with what we see in this part of the trial. So the next slide. So this is, as I mentioned, we have this potential of bilirubin evaluation. And bilirubin is a standard procedure in the hospitals, so it can be done without any specialty. And what we know from the mode of action of SCO-101, it could increase the bilirubin, which is a liver enzyme, or it's generated in the liver, and it could increase and then decrease after a few days. And we can define or subdivide the patient population into 2. Those patients who had a rapid increase in bilirubin and then a decrease thereafter, and those where it increased but stayed high. And these that had stayed high, they are called permanent or persistent increase, which we'd also define this as high bilirubin index. And conversely, those that increased and then decreased very rapidly, these are the transient and have a low bilirubin index. Again, the Kaplan-Meier plot on the left-hand side shows the overall survival of subdivided based on the bilirubin index. And as you can see here, there is a significant increase in overall survival for those patients with a transient bilirubin index, amounting to about 13.4 months. So this is very, very good data. And the next slide, please. So now we're turning our attention to CORIST Part 3, which as I discussed earlier, was based on an optimizing dosing evaluation. And here one of the main take-home messages that we can see already now is that we saw 2 patients. It may not sound a lot, but we saw 2 patients with a partial remission, which means there's more than 30% reduction of the tumor. And I'll show you an example a bit later on. And this is so interesting, and also Professor Tabanero was mentioned this specifically, because we know that in this line of therapy, the response rate is 5% or less. And in the recent, as I mentioned earlier, the Sunlight trial, this was a study with more than nearly 500 patients, and in that patient population, which are in the same patients that we have in our CORIST Part 3, the response rate was 6.1%. So they had 15 responders out of the 246 patients treated in that arm. So again, very exciting and promising data. We also saw a very good clinical benefit rate, and we saw a promising overall survival and progression-free survival. Now, we also looked at this part of the trial, and this is also something that Tabanero was really highlighting. This was a UGT1A1. Yes, I did say it right. Sometimes it's difficult really to all these numbers, like saying dinosaur names. However, we saw that that in this patient population with a wild type, and I'll show you the data a bit later, the median overall survival was 13.7 months. And the next slide, please. Before I show you this, I need to highlight this slide, because as also Jens said, we with our drug can increase the exposure of SN38. SN38 is the active drug of irinotecan. So here on the gray curve is what we see with the PK profile of SN38 or irinotecan when it's given without SCO-101. And you can see in the blue curve what happens when we add SCO-101. And we get a 4 to 5 higher increase in exposure. And therefore, we have, and preclinically, we have shown that we can also increase it in the cancer cells. We cannot show this in the clinical setting, but we assume there's a parallelity between non-clinical and clinical. So we have a higher exposure for a longer period of time with a potential of having higher intracellular SN38. This is a win-win situation for us, and it's a cancer cell's worst nightmare. Now, next slide. So this is looking at the UGT1A1 genotype. And I would like to highlight, we have looked at this from various points of view, but the solid line is the normal expression of UGT1A1, and the 2 other lines are various types of abnormalities. And I would just like to highlight that we do see in that patient population which have a wild type UGT1A1, the overall survival is quite impressive. Well, I have to say that this can increase further, in as much as some patients are still active on treatment and alive early on in this curve. So the curve could, or the overall survival could increase over time. And the next slide, please. Now, turning to real life data. For those of you who are not used to see the CT scan, I'll come back. I'll show you more. I'll walk you through it in a minute. But these represents a 55-year-old female with a RAS mutated colon cancer, which was diagnosed in 2019. She entered the CORIST trial in August 2023, so part 3. on the highest dose level, so 250 milligrams of SCO-101, and she is still alive and still being treated. So on the left-hand side of the CT scan on the left-hand side, this shows the baseline in August 2023, and circled in the blue line is the tumor mass as it represented in the left armpit. At that point in time, after 16 weeks on treatment, you can see that the tumor is not completely disappeared, but it's very blurred and it's shrunk a lot. So therefore, the radiologist at this site measured this to be 10 millimeters, and so this is a partial remission. So the patient has been scanned several times thereafter, and it's still a partial remission, and she's doing very well. I talked to the principal investigator a few days ago, so everything is going very well for this patient. And the next slide, please. So, what are our next steps? We would like to establish the maximal tolerated dose of SCO-101 in the 4-day schedule. And we would like to do this by increasing the dose of irinotecan to 65 or 80%. First 65, and if that is tolerated, then increase it to 80%. The percent of what? Because this is a percentage of what is the normal dose of irinotecan that's given with FOLFIRI without SCO-101. We would also like to evaluate the efficacy of SCO-101 with FOLFIRI, and we'll do this by looking at remission, overall survival, progression-free survival, and clinical benefit. We already have these data from the first patients, but we'd like to extend that data package and also look at further into the UGT1A1 expression and see how that correlates as a potential biomarker. And the next slide, please. Now, as also Professor Tabernero said, our ultimate goal is, of course, to move from the biological proof of concept into the clinical proof of concept, and this can only be done in a randomized clinical controlled trial. We have very recently had the ESMO guidelines. So ESMO is the European Medical Oncology Society that gives guidelines for the standard treatment for patients with malignancies. So the ESMO guidelines from 2024 dictates or stipulates that there is a new standard of care which is long-served with bevacizumab. This is based on the Sunlight trial that I alluded to a bit earlier. So we propose that we will have a randomized clinical trial in the third line of patients with colorectal cancer. We will be using FOLFIRI with SCO-101 at that dose that we have established from CORIST Part 3. And then we will evaluate this up against the best standard of care. And we are still working with external experts, US key opinion leaders, and we'll also be discussing this further with our European key opinion leaders. We will ultimately also be discussing this with EMA and the FDA in order for us to get a handshake on the clinical development program. Now, I also in the bottom left on this slide are given a potential timeline, so the recruitment time, 1.5 to 2 years, and the efficacy readout about a year. Now, these may sound very long timelines, but we have, of course, a possibility to look at something earlier, like clinical response rate, clinical benefit rate, progression-free survival, etc. So we will have some indications earlier on, so we don't need to wait for these nearly 3 years before we have the final data. So on our next slide, my last slide. So these are the take-home messages that we have from CORIST Part 1, 2, and 3. We have shown that we have a very good safety and tolerability profile. We have seen 2 patients with a partial remission amongst the 6 patients in the last cohort. We have seen impressive overall survival across the different sections of CORIST between 8 and 10 months. However, we have also seen that we can subsegment the patients based on the UGT1A1 And in that patient population, we increased the overall survival from 8 to more than 13 months, which is quite significant and also of clinical relevance. Now, our positive data warrants expansion of CORIST Part 3 by adding a small patient cohort to further optimize the dosing schedule and also the dose of irinotecan to be applied in a later randomized clinical trial. So my final message is, overall, our data is very, very encouraging and supports our continued development strategy of SCO-101 in combination with chemotherapy. And with that, I'll hand it over to Johnny.
Thank you, Lars.
Thank you.
And I will just quickly take you through the rights issue as we have announced. We are doing a rights issue here in the second quarter of this year. So I'll just take you through some of the key dates and the structure of the rights issue. So the rights issue will comprise units. As a shareholder, for each unit, you will receive 3 things. You will receive 1, 4 shares, 2, 3 warrants in a series of what we call TO2, and 1 warrant in the one series called TO3. For the first one, the shares, the subscription price of the share will be set on the average share price, based on the average share price from the period of May 17th until May 23rd, where the price of the share will be calculated. As mentioned, number 2 and 3 consists of warrant structures. So the first warrant structure, TO2, that consists of the 3 warrants, and they are to be executed in the 4th quarter of this year. So the price will be set in the second half of October 24, and subscription of the warrants will take place from November 4th to November 18th, so the first half of 24. The second warrant, the TO3, will take place in the second quarter of 2025. Now the price will be set in the second half of March 25, but the subscription will take place in April of 2025. Some key dates to be aware of: the publication of the final terms of the rights issue, including the subscription price, as I mentioned before, that will be announced on May 23rd, followed by the prospectus, which will be published on May 29th. Following this, going into June, we will have some key date intervals to be aware of. The trading in the unit rights, that will commence in the period from June 4th until June 17th, in which period you can trade the unit rights. In the same period, and going an additional 3 days from June 4th until June 20th, that's the subscription period. If you wish to subscribe for shares in the rights issue, you can do this in this period, and you can do it through your bank. So reach out to your bank and they will guide you through how you use your unit rights to subscribe for shares. Thank you. With that, I will give the word back to François for closing remarks.
Thank you very much, Johnny. And before we will open up the floor to any questions you may have, I thought I would summarize in a few slides where we stand and where we will be going to. So you see on this slide, this is really the pipeline of our company, and you see on the left-hand side, and first of all, I would like to mention pancreatic, because we released very recently some good pancreatic phase 1b data, mentioning good safety profile, good tolerability profile, and that was one of the milestones for this year. Then we have released earlier the CORIST Part 3, as Lars was mentioning. We will be waiting for the final analysis of the CORIST Part 3 a bit later in the year. That is expected to be in H2 of this year. Now, what we will be doing, the next couple of months is what we call CORIST Next Step, CRC phase 1b/2a. We will be starting to enroll patients as soon as the right issue will be over, as soon as we have approval from various committees, and therefore we will expect to enroll patients, up to 12 patients, a small cohort of patients during the year. And then hopefully we will have the top line result around year end, around the end of this year. And then we will be having a major data readout during H1/H2 25. So this is what exactly we will be doing in the next couple of months at Scandion Oncology. It means also that we will not be pursuing gastric phase 1b/2a, nor pancreatic phase 2a, not because we don't think it is appropriate to develop those indications further, but because we need to prioritize our resources for colorectal cancer. Right, so to sum up where we stand as a company, I hope that Jan, our CSO, and Professor Tabernero as well, were able to convince you that we do have a potential first-in-class drug inhibition company with a new targeted dual mechanism of action. And we are one of the first company, in cancer drug resistance, as Jan was mentioning. We are working in indication with a very high level of unmet medical needs. I wanted to summarize this very quickly. When we diagnose, when colorectal cancer is diagnosed, it's very often at a metastatic stage, and unfortunately, the survival rate is around 12% at 5 years. So there is obviously a broad commercial potential of this drug. And then we, as Johnny was mentioning, we will be continuing to control our costs, certainly throughout the year and beyond. And the right issue will allow us to continue to fund operation, to perform this study I was mentioning before. We call that CORIST Next Step, or the Bridge study. And we will be able to continue funding operation into H2 25. Now, I hope that we were able to convince that we have a very strong focused pipeline and clinical development as well. And all in all, that we all are very senior level executives. We're having, I haven't calculated, but it's probably over the 100 years of experience altogether. And we have, as I mentioned before, we have added to our board, 2 very senior level clinical oncologists, bringing, one and the other different skills and experience, on a European level. So the value inflection point for 24, CORIST Next Step study, as I mentioned, and the final CORIST Part 3 results towards the end of this year. Having said that, I would like to thank you, you shareholders or potential investors, for your continued support and trust in our company. And having said that, I will pass the floor back to you.
Thank you very much, Francois, and of
You're
course
welcome.
the rest of the management at Scandion. I thought I wanted to tackle the rights issue first. Should we invite Johnny back to the stage?
Right?
Yeah. So first and foremost, why does Scandion need to have a rights issue currently?
As we have announced, we have extended our current runway quite significantly to go through last year and into this year through cost-saving measures and restructuring of the company, because we saw that the market were as difficult, the funding markets, as they have been for quite some time now. We have now reached the point where we do need to secure additional funding to progress the company and to progress the CORIST study. as mentioned by Francois, we will be using the new funds to further progress the CORIST with this next step study.
I saw in the plan that there was many different sort of rounds of capitalizations in this rights issue with the rights that the investors get. But allow me to ask that when at full coverage, could you explain how exactly you expect these funds to be allocated throughout this time?
So we, from a cautious point of view, we base our progress and our budget on the amount which is guaranteed. So as we have informed and announced, that the guaranteed minimum amount will enable us to do the next step study and progress the company to the point, as Francois mentioned, into 2025. Should we get, and we hope so, to getting additional funds, that will enable us to progress further and start the initial work of preparing for a randomized study in CORIST.
So the current budget and plan is based on the current guaranteed volume of the rights issue.
That is correct.
But like you were in a little bit, the rights issue is secured. It's 51%. Can you explain a little bit on who is backing you in this rights issue?
Yes. So management and the board of directors, they are all participating in this round. And on top of that, we have two groups or two guarantors. So it's very positive that we have been able to secure 50% with a very few number of guarantors, which has shown a strong support to the rights issue.
And should the subscription rate sort of reach above 51%, can investors expect updated roadmaps and budgets?
Well, of course, as we progress and depending on how the subscription of the shares goes, and then further on at year end or Q4, we have the first round of warrants TO2, and then further into Q2 of 25, the small round TO3. Depending on the outcome of that, of course, we will announce to the market what the outcome have been of those 2 free events and what that means for the company. We certainly will.
Thank you much, Johnny
Thank you.
I will also have some questions for Jan Stenvåg, who is Chief Scientific Officer. Thank you very much. We were talking about the sort of science behind the SCO-101, and I'm curious, the mode of action, what is unique about it?
Well, the unique thing is that we have a simultaneously targeting of 2 different key enzymes in resistance, and it seems to be quite specific. So the uniqueness lies in that mode of action, you could say, and also that no one else has this mode of action. So that makes us unique, we think. Yeah.
I'm by no means scientific myself, but the graphics you showed about the sort of plugging the holes in the chemotherapy treatment, I thought it was very informative. And I'm curious, when you're developing this for, say, commercial use, do you see it more like targeting the chemotherapy therapy itself, or do you see it targeting specific forms of cancer one by one?
No, we think it targets more the specific chemotherapy. So that means eventually it could be broadened out to other indications, such as gastric, as Francois alluded to, which also irinotecan is used. So obviously we think that based on the mechanism of action, it could potentially be broadened out to a number of other cancer indications, potentially with more than irinotecan, also with other types of chemotherapy, yes, clearly.
And how are you exploring that sort of breadth?
That is done in several ways. First of all, we definitely talk to our key opinion leaders and ask what is the medical need, what is used of chemotherapy, and what data do we have to back up that this chemotherapy would be relevant. Then we look into the literature, of course, which clinical trials have been done, how big are they, what was the outcome, what is their position for Scandion Oncology to tap into that market. And then, of course, we make some initial market analysis based on external assistance, obviously, to see how does it all fit together. So obviously we do that. But as Francois said, right now we have a very strong focus on the colorectal cancer.
Thank you very much, Jan. I also have some questions for Lars.
Thank you.
Yes. So thus far, how many patients have been treated with SCO-101?
So if we start with Pentax, it's 22. For CORIST, it's 68. So totally 90 patients. On top of this, we also have a number of healthy volunteers. And of course, they're not patients, but in total, about 150 individuals have been treated with SCO-101.
And moving on, when you do the adding the patient cohort and as well as moving towards phase 2B, how many can you expect to be treated?
First of all, for CORIST, so extension or the next steps, we plan for up to 12 patients. In order for us to get the maximal tolerated dose. With respect to the phase 2B, I cannot say at this point in time, because that of course depends on the number needs to be treated, which depends on all the statistics, and I'm not a statistical expert, so I can't say, but so I will not even go down that route.
All right, maybe down the line then.
Yes, down the line,
Yes.
I will come back and tell you.
Of course, but tell us more why you need to do this extension study and why not start the randomized phase 2B right now.
First of all, and this is also the feedback we had from key opinion leaders, we at the moment, in the CORIST part 3, we're using 100% dose of 5-FU, so one of the chemotherapeutics. We're using 100% of folinic acid, but we're only using 50% of irinotecan. And they would like to see us increase that dose to get more to the 100% dose. And this is why we want to explore the 65 and 80% of the normal dose of irinotecan to push it even further, because we hopefully we will see additional efficacy benefits from that without having extra toxicity.
So it's about finding this sort of optimal dose.
Yes, actually, the goal is to find the maximal tolerated dose, which of course then needs to be implemented as a recommended phase 2 dose in the randomized clinical trial.
And as you move forward towards the randomized phase 2B study, what sort of preparations need to be made? We were talking about how many patients you were expecting to sort of treat with the SCO-101. Is that part of it, sort of managing how many patients?
First of all, appreciate the question, and it will take all day to explain in detail, so I'll just do the highlights. First of all, we need to have interactions with our key opinion leaders. And then we need to have an interaction with the biostatisticians to evaluate how many patients do we need in these various, in the 2 arms. And based on that, we will then go to FDA. We could go to FDA and have what's known as an IND. We could go to EMA and have a discussion with EMA about this design. And ultimately, we need to have a protocol, the cookbook for the trial.
Thank you very much. So, Francois, I think I'll end with you then. So I wanted to talk about a bit of partnerships. What do you look for in a potential partnership?
Well, you know, as a small company like Scandion, we are always discussing with potential partners, meaning, you know, big biotech companies, small-size pharma, mid-size pharma, big pharma. So it's an ongoing dialogue that we have initiated a few months ago, and of course we will continue to do that and strengthen it down the road as well. Absolutely.
Is there any discussions currently going on?
There is discussion ongoing, absolutely, but you can easily understand that I cannot tell you a little bit more about that.
Of course, of course. I'm curious to know what sort of aid you expect from a partnership. Is it financing, or is there more?
Yeah, I mean, clearly, I mean, as Lars was mentioning, you know, the phase 2B randomized, that could be also a phase 2B/3, would require a significant number of patients to enroll in the order of magnitude of 400, 500. So therefore, this will imply a significant cost for the company. And that's the reason why, fundamentally, we're looking for a partner in order to help us to perform that study.
The additional patient cohort here for the CORIST study, I'm curious if, say you get very positive results from it, how will that change your scope for partnerships?
Well, that certainly will increase the scope of it. Absolutely. So again, just to summarize, so this small study is done for MTD, maximum tolerated dose, but also we will be looking at efficacy, so potential partial responses. And as you said, having done both, then we could certainly be even more exposed to potential partners. Absolutely.
Thank you very much, Francois.
You're welcome.
I will open up the floor here for any questions, and I will also take a question that has come from the chat. How do you expect to finance the 2B randomized study?
I just said,
I
mean, clearly, we would need to have a partner in order to be able to match, the financial requirements of that study. Absolutely.
And any questions from the crowd? No, all right. Then I wish to thank you, Francois, and of course the rest of the management team at Scandion. Thank you very much.
Absolutely. Thank you very much. Thank you.